Hello guys, I hope you all are doing well at home and enjoying our online lecture videos also. So I again request you to subscribe our channel NUCLEIN Biotech by pressing the bell icon given below for more such videos, and also don't forget to like, comment and share. Now our today's topic is somatic hybridization. So before proceeding to the introduction of somatic hybridization, let's understand the meaning of this term. This is also known as somatic cell hybridization. So here somatic cell means those biological cells which help in forming the body of an organism, or we can say cells which can differentiate. So here soma means body. Next is hybridization, means the process of an animal or plant breeding with an individual of another species or variety. Now let's move on to the definition of somatic hybridization. It involves in vitro fusion of isolated protoplasts to form a hybrid cell and its subsequent development to form a hybrid plant. So when the cell wall is removed from a cell, then we get protoplast. I think we all know about this. And in vitro means any process performed in a test tube, culture dish or elsewhere outside a living organism. Now hybrid means mixed characters, and hybrid plants is the result after hybridization, and it provides a novel opportunity to create cells with new genetic constitution. Now move on to the steps involved in this process. There are 3 steps. First one is the fusion of protoplast, second one is the selection of hybrid cell, and third one is the identification of hybrid cell. Let's start with the fusion of protoplast. In this, isolated protoplasts are devoid of cell wall, so in vitro fusion becomes easily as there is no barrier of incompatibility for the protoplast fusion. It can be achieved by 3 methods: spontaneous fusion method, mechanical fusion method, and induced fusion method. Let's discuss about these methods one by one. First is the spontaneous fusion method. So in this method, cell fusion is a natural process as it observed in case of egg fertilization. During the course of enzymatic degradation of cell walls, some of the adjoining protoplasts may fuse to form homokaryocytes. These fused cells may sometimes contain high number of nuclei that may vary to 2 to 40. The frequency of homokaryons formation was found to be high in protoplasts isolated from dividing cultured cells. Spontaneously fused protoplasts, however, cannot regenerate into whole plants except undergoing a few cell division. So here is the word homokaryocytes, which means cells which are multinucleated. Next is the mechanical fusion method. In this, physical fusion of protoplast, or we can say protoplast, that can be pushed together mechanically to fuse under microscope by using micro manipulator and perfusion micropipette. But the drawback of this method is that it may damage protoplast by causing injuries. Now next is the induced fusion method. In this, isolated protoplasts are fused by fusogens. Fusogens are nothing but the fusion inducing agents, for example NaNO3, high pH, high calcium ion, PEG, which we gonna study further. Now first is the treatment with NaNO3 or sodium nitrate. This is first reported by Power et al. In 1970. In this, isolated protoplasts are cleaned by 10% sucrose solution and then transferred in 5.5% of sodium nitrate solution. After that, incubate for 5 minutes and then centrifuge it again for 5 minutes at 2000 rpm. In the last step, these centrifuged protoplasts can kept in a water bath at 30 degrees Celsius for 30 minutes, during which fusion occurs. This treatment results in low frequency of heterokaryon formation. Now next is the high pH and high calcium ion treatment. In this, different protoplasts are treated together in a solution with condition having high calcium and high pH so that they can fuse together. In this, isolated protoplasts are incubated in a solution of 0.5 mole mannitol containing 0.05 mole calcium chloride. Here, pH should be 10.5 and temperature approximately 37 degrees Celsius for 30 to 40 minutes. After this, aggregation of protoplast and fusion usually occurs within 10 minutes. By this method, 20 to 25% of protoplast involved in fusion. Now next is the PEG treatment. PEG means polyethylene glycol treatment. This is one of the most effective methods for protoplast fusion. Isolated protoplasts in culture medium are mixed with equal volume of 28 to 56% of PEG in a tube, like 1 ml culture medium and 1 ml of PEG. After that, tube is shaken for 5 minutes and allowed to settle for around 10 minutes. At last, settled protoplasts are washed several times with culture medium. This is widely used method, and the advantages of this method are high frequency of heterokaryons formation and the low toxicity to cells. Now last step is electrofusion technique. In this fusion induced by electrical stimulation, again in this process, first low voltage electric pulses are passed in the solution to form a pearl chain of protoplast, and then high voltage electric current is passed, which leads alteration of membrane the adjacent protoplast fuse. Low electric current should be 10 kilowatt per minute and high electric current is 100 kilowatt per minute. After the fusion of protoplast, we get a solution having three types of cell. One is unfused protoplast of two species, and second is product of fusion between two or more protoplast of the same species, and hybrid protoplast produced by fusion between one or more protoplast of each of the two species. So there are two methods for selection of hybrid cells. First one is the biochemical method, and second one is the visual method. Now let's discuss them one by one. Biochemical method is based on the use of biochemical compound in the medium, that is known as the selection medium. These compounds help to sort out the hybrid and parental cells based on their differences in the expression of character. Again, it is divided into two methods. First one is the drug sensitive section method, and another is the auxotrophic mutants. Now in drug sensitive selection method, this method is useful for the selection of plant hybrid species if one of them is sensitive to a drug. Here are the two species of same plant in which species A can form microscopic callus on MS medium but are sensitive, or we can say inhibited, by actinomycin D, and species B can form small colonies but are resistant to actinomycin D. When these species are fused together, the fused protoplast shows both the characters formation of microscopic colonies and resistance to actinomycin D on MS medium, and this helps in selection of hybrid. Now next is auxotrophic mutants. Auxotrophs are mutants that cannot grow on minimal medium and therefore require specific compounds to be added to the medium. For example, nitrate reductase deficient mutants of tobacco are known. The parental protoplast of such species cannot grow with nitrate as the sole source of nitrogen, while the hybrids can grow. Two species of nitrate reductase deficiency, in which one is due to lack of apoenzyme NIA type mutant and another is due to lack of molybdenum cofactor, that is CNX type mutant. So the parental protoplast cannot grow on nitrate medium while the hybrid can grow. Next is the visual selection method of hybrid cell, in which is very tedious but is very efficient. In some of the somatic hybridization experiment, chloroplast deficient non-green protoplast of one parent are fused with green protoplast of another parent. This facilitates the visual identification of heterokaryons under light microscope. The heterokaryons are bigger and green in color, while the parental protoplasts are either small or colorless. Further identification of these heterokaryons has to be carried out to develop the specific hybrid plant. Now again there are two approaches. First one is visual selection coupled with differential media growth. These exist certain natural differences in the sensitivity of protoplast to the nutrient of a given medium. Thus, some media can selectively support the development of a hybrid but not the parental protoplast. Now second is the mechanical isolation. The visually identified heterokaryons under the microscope can be isolated by mechanical means. This involves the use of a special pipette, namely Drummond pipette. These so isolated heterokaryons can be cloned to finally produce somatic hybrid plants. The major limitations of this method is that each type of hybrid cell requires a special culture medium for its growth. This can be overcome by employing micro-drop culture of a single cells using feeder layers. Now move on to the identification of hybrid plants or hybrid cell plant. Now in this, first is the hybrid identification. It is based on difference between the parental cells and hybrid cell with respect to pigmentation, cytoplasmic markers like like FITC and RITC are used for labeling of hybrid cells. Now, presence of chloroplast, nuclear staining. Heterokaryon is stained by carbol-fuchsin, acetocaramine or acetoocin stain. Next is the hybrid selection. There are several markers are used in this method, like genetic complementation, phytotoxins, specific amino acid, auxin-autotrophic antibiotics, auxotrophic and metabolic mutants, chromosomal analysis, and herbicides. Culture of the hybrid cells on suitable medium provided with the appropriate culture conditions. Now, next is the regeneration of hybrid plants. Plants are induced to regenerate from hybrid calli. These hybrid plants must be at least partially fertile in addition to having some useful property to be of any use in breeding schemes. Now that's all for the today's topic. If anyone having any query related to this topic, they can comment in the comment section. Thank you.